Hi everyone, my name is Abraham Sebek, and welcome to SAT 2 Biology. Okay, so this is lecture number 1, and in this lecture we're going to go over the introduction to biology, okay, because the whole point of taking the SAT 2, right, is to get an 800 on it. We want you guys to get the highest possible score so you'll have your pick of colleges and your pick of educational programs, right? So I'll start this lecture off by going over the fundamentals of life and with the unity of life. There's so many factors that unite all living things together. Okay, so let's start with the first thing. And this is the life processes. Do you know all the life processes? Growth, nutrition, respiration, regulation, reproduction, excretion, synthesis, and transport. The way I memorized it was I would just remember a good night's rest. Okay, so just very simple, good night's rest. All right, and then what I did was briefly just put, so a good student always gets a good night's rest. All right, and so briefly I put what each of these life processes are all about. Now, every living thing, coming from the bacteria to the human, has to be able to do all of these life processes in order to be deemed alive. Okay, and so this looks really nice, all right? Growth is the number of cells. Now, nutrition is either autotroph or heterotroph. Very simple. With the autotroph, all we need to remember is that they are self-feeders, okay? So they do not require, they're self-feeders. They don't require any food, right? They can feed themselves with photosynthesis. Heterotrophs, okay, these guys are consumers, right? These guys need to eat something to stay alive. All right, respiration is gas exchange. Right? Also, we're going to make ATP using this gas exchange. Regulation is responding to the environment, like the endocrine system, or using hormones, or the nervous system using neurotransmitters across the synapses. Right? Reproduction is just sustaining this species, and it could either be sexual or it could be asexual. And then excretion is waste removal. Living things never get to use all of the nutrients that they take up, or all of the particles that they take up. And so there's always going to be some sort of waste product. Even if it's a gas, you still have some sort of waste removal. Synthesis is when we build very large molecules, like for instance DNA, RNA, proteins. These are very long polymers, and we're going to see that when we do dehydration synthesis, right? Cells do dehydration synthesis. And finally, transport. I put circulation here, but that's even at the cellular level, where the cell can circulate its cytoplasm around. It's able to move its nutrients from one location to another, really. That's what transport's all about. And the endoplasmic reticulum in the cell plays a big role in terms of transport. Okay, so the next thing we have to know is the hierarchy of life. And a nice, easy way to look at this, I know it looks like a lot, but you're going from the largest item up here, and then we have our smallest item down here. Oops. Okay, and biosphere comes first, all right? So this is like the Earth. And then going even smaller, we have an ecosystem. An ecosystem has all the living and the non-living things together, okay? While the community only has the, so over here I'll put non-living, right, and living, all in one. Right, but then the community is only going to have the living things. The population is going to be the same species. An organism, okay, so think about a human being the organism. He's made up of many organ systems, like the respiratory system and the circulatory system. The organ would be the heart. Right, going further, the heart is made up of 4 different tissues. Epithelial tissue, we have connective tissue, we have muscle tissue, and we have nervous tissue. The cell, okay, the cell they are made up of eukaryotic cells, animal cells, right? Going even further down, we have organelles. These are the little organs of the cell. Organic molecules like carbohydrates, lipids, and proteins. And then finally we have our molecules, very simple ones like water, methane, ammonia, and we have our atoms. Okay, we're going to see more of this when we go into our chemistry unit, but for now, just keep in mind this is the largest and this is the smallest. Okay, ecosystem has both the non-living and the living factors all in one. The world is made up of many, many ecosystems. Okay, and in some ways, maybe we can even put the word biome in between. All right, so let's get this out of here. So you got to know the hierarchy of life. So what's up here is very important. they'll sometimes ask you on the SAT too, like, what is the correct order? And they might go from cell to tissue, organ system, organism. It's very common that they'll do that, arrange in the correct order. And so you definitely want to know the order that it comes in. Okay, speaking of which, we have the types of cells here now. There's two major types of cells. One are prokaryotic cells, while the other are eukaryotic, okay? If they're prokaryotic, they don't have a nucleus. These have DNA. No matter what, you have genetic material. But in this case, you don't have a nucleus to actually keep your DNA wound in there, okay? It also has one single chromosome. In fact, this region here, it's like a wannabe nucleus, right? I call it the wannabe nucleus. This is the nucleoid region. Okay, so bacteria and prokaryotes, okay, in the monera kingdom, they have just this nucleoid region. And that's what's the major difference. Also, they don't have organelles with membranes. For example, the endoplasmic reticulum has membrane, right? The chloroplast, the mitochondria, the Golgi complex, the peroxisomes, none of those things exist. Really, all they have is cytoplasm, some ribosomes, and their DNA. So they're really, really simple. On the other hand, the more complicated cells are called eukaryotic cells. These cells have a nucleus, they have more than one single chromosome, and in addition, these are the kingdoms that we'd probably find with eukaryotes, right? Animal cell, plant cell, fungi cells, and protist cells. They all have a nucleus, and they all have membrane-bound organelles, like the endoplasmic reticulum, the Golgi, the chloroplast, the mitochondria, the peroxisome, etc. Okay, so this is really important. Make sure we know both types of cells, because they're going to use these words like it's nothing, prokaryote and eukaryote. If you have no idea what those words are, you're going to be at a serious, serious disadvantage. So make sure you know them. Okay, another thing is genetic material. Every living thing has DNA in it, right? If you're alive, you must use the DNA code that uses 4 bases, right? And here are the 4 bases. We have thiamine, okay, we have cytosine, we have adenine, and we have guanine, right? So we could just put them over here as T pairs with C, right? And A, well actually that's not correct. A pairs with T, and C pairs with G. Okay, this is very important that we know that this always happens, right, where the base pairing is happening between the two between the two nitrogen bases. Okay, one nice way to remember this is the purines. Notice how they have 2 rings. Okay, if they have 2 rings, we'll just easy to remember purines. 2 rings. Purines. We used to walk home and remember that. 2 rings. If you can remember that, and then also pure as gold was another thing that helped us out. So if you remember pure as gold, then you just remember that purines include adenine and guanine. Okay. Now, if we're looking at up here, uracil would be the other one, so I could put U right there. That would be an RNA, right? You'd have like instead of T, this would be only DNA, T, and the thymine. You could just remember what? Cut the pie, right? So cut the pie. So remember that pyrimidines include what? We have the cytosine, the uracil, and the thymine. All right, cut the pie. Cytosine, uracil, and thymine. Very easy to remember. Pure as gold. And you know your bases, and you're well ahead of everybody else in terms of knowing these properties. Okay, remember they base pair. They base pair using hydrogen bonding. All right, and let's continue. So we know every living thing must have genetic material. Now, the next thing we should look at is how do we classify? So this is like fancy word right here, taxonomy. It's just a fancy way of saying classification. Classification is where we group organisms into similar groups, right, based on their properties. So this is just an example of, this is Carlos Linnaeus, right? Linnaeus came up with a nice taxonomic scheme. Domain is fairly new, and we're going to go over that as we go on later on when we do the taxonomy unit. But for now, starting with kingdom, we're the animal kingdom, right? I'm looking at this cat over here. He's a chordate, which means, chordate means you have a backbone, or you're vertebrae more or less. Mammalia means that you give milk, right? You're a warm-blooded organism that's giving milk to the young. Carnivore means that you eat meat. Felidae is the cat family. Panthera is the genus, and Panthera pardus is the species. And so can we do the human? let's try to see if we can put the human in this taxonomic scheme that Carlos Linnaeus has set up for us, right? So human would be, same thing, this would be the animal kingdom. And then also chordate would be our phylum. Mammal, right? We'd also be a mammal. Carnivore, no, we'd be primates. Okay, so this is where we start to differ with our panther friend over here, right? Felidae wouldn't be our family. We actually belong to something called hominidae. Okay, a good clue is notice how they end in day. Usually the family always ends in D-A-E with that suffix, okay? Homo is the genus, and sapien is the species, okay? And these last two make up the scientific name, right? Our scientific name, whenever we would just probably say Panthera pardus, Homo sapien, Felis leo, and so forth. Okay, so let's move on. Let me get this out of here. Next is evolution. So this is another underlying theme that every living thing has. They change over time, right? Evolution is just changing over time. And who should you know about? This guy right here. That's Charles Darwin. Okay, Charles Darwin is the father of evolution because he comes up with the theory of natural selection. Now you got to know the 5 points. I've seen this so many times on the SAT too, it's not even funny. So you better memorize these 5 points. Overproduction, right? Meaning that you have way too many offspring. And then, of course, in sexually reproducing species, variation would exist, right? Because of so much crossing over, so much meiosis. Every organism looks different from the other. Okay, limited resources, meaning that there's not enough food space, there's not enough mates, there's not enough shelter to go around, and therefore competition between members of the same species will occur. And then, of course, survival of the fittest. Now, this word fittest is so important. Fittest just means whoever has the most offspring, okay? I can't stress that enough. I always tell my students it's the most offspring. It doesn't matter if you're the healthiest. It doesn't matter if you're the strongest, the fastest. You have to have the most colorful, just most offspring. Whoever gets to mate the most and produce the most is the fittest. There it is, okay? And so this is very important. This is another thing I wanted to show you. Darwin went on the Galapagos Islands, right? And he observed the finch, these tiny little birds, about this size, yay big, right? And they would have beaks. He noticed that their beaks were different from island to island to island. It all depended on their food source, okay? And so that would determine whether or not they were actually going to have this type of beak. Okay, and then finally the giant tortoise. So this thing is huge, and in England they never had anything like this, right? He's from England. And he noticed this also on the Galapagos Island. And stuffed him and brought him back. Found 2 species, right? One that can eat bushes, right? And another one that didn't have that long neck or that opening. They had like an opening right here, and then instead the other one had a closed opening and he had to always eat off the ground. Actually, that was the availability of food on the island, and so this worked out really nice. Okay, for the tortoises, seeing the variety out there, seeing the different species, and indeed that evolution does exist. Okay, next is the scientific method. First day of school, they always give this to you. I don't want to spend a lot of time on this. However, you have to remember that you start off with an observation before you come up with a problem. Problems always arise by you noticing something. Flashlight doesn't work, just like in this problem, okay? Then you should figure out how flashlights work, or exactly the underlying thing about your, like the research is saying, did anybody else ever test this problem out? Has this already been solved? Okay, because we're not going to do an experiment about something that somebody already drew a conclusion to. A hypothesis, okay, now this is where we come up with a theory, not really a theory, but a prediction. Okay, we're making a prediction. Well, what exactly is the reason for this problem? Maybe the batteries are dead, okay? Then we have an experiment, let's test it out. Let's try putting the batteries in and changing them and seeing if the light bulb or the flashlight will still light. You take your data down, did it work, yes or no? And then that conclusion, the conclusion should be attached to the hypothesis, okay? They go together. And that's where in your conclusion you would start to talk about whether your hypothesis is rejected or accepted. Now on the SAT too, they always bring up different experiments. So I'm going to take some time out with these lectures and give you different experiments to look at. Just the highlighted ones that are very famous and that every good biology student knows that almost every single textbook goes over. All right. And so let's continue on, but remember, they do have sections where they devote 5 whole questions to just not scientific method, but an actual experiment. All right, the last thing I want to go over is designing a controlled experiment. So just real quick, the control is something, they'll sometimes ask, which of the following serve as control or controls? All right, this is the part of the experiment you do nothing to. Sometimes an experiment will test more than one variable, okay? So they'll have like a multi-variable experiment. Well, in any way, variable is something that you change, okay? So there's 2 things that we're going to notice that change during the experiment. The independent variable is the part that we change. Okay, the part that the person who's the experimenter or the scientist changes. The dependent variable is what we're trying to measure. So let's say we wanted to find out the amount of, or the what effect does the color of light have on plants, right? We'll want to measure, so the thing will change is the wavelength of the light. But the thing we're going to measure is the plant's growth. Okay, next, constants. Constants are not being tested and they should stay the same. Things like pot size, soil, the species of plant, the amount of oxygen in the room, the amount of water, and so forth. The sample size, how many number of organisms do I use in this so-called rainbow experiment where I'm taking different colors and testing them on plants. Right? How many plants will I use? That's the sample size. All right, and with that said, this goes over every single thing that you need to know in terms of the beginning of biology, and so next time we'll look at the chemistry behind biology, okay? Just until next time, start taking maybe a diagnostic test or a practice test and give it about an hour a night, and you'll start to see your SAT score really increase. Until next time, I'll see you. Be safe. Hi everyone, my name's Abraham Sebek, and welcome to SAT2 Chemistry. Okay, so in these lecture series, we're going to be doing all the problems for SAT2 chemistry and go over each and every one of the topics. So we can prepare you for the test and also get you an 800 top score on the test. So first, let's go over the format of what we're going to see on the SAT2 to better prepare you. And the first thing is there's 85 questions on the exam, and all you're given is 60 minutes. So you have about 42 seconds per question. So you have to keep moving, okay? So your knowledge, being like swift with chemistry and being able to just remember everything, that's gonna be really, really important. So these lectures are designed to do that for you. Next, the matching questions, okay? So the first part you get is 25 questions. They're all matching. So they'll give you, like, for example, a few words, and then they'll give you a short statement that you have to match it with. 15 of the questions will be true and false, where they'll give you 2 statements, and you have to decide whether the first statement it correctly explains the second statement. All right, and this is unique only to the SAT2 chemistry, and they're numbered from 101 to 115. Then you have your multiple choice questions, okay, and that's a total of 45 questions. So the goal here is to maybe omit 5 questions the most, okay? I tell students all the time, if you have to, don't go over 5 omits. I would probably go 4 or 3. I've talked to people who've gotten 800s, okay, I myself have gotten an 800, and I remember not going over maybe 4 omits, if anything. And I made sure that I got these questions all correct, and then these are the ones where you want to save the omits for, if anything, unless you're totally clueless about any one of these, which you don't want to be. Okay, so the next thing is they give you a periodic table. You're given no calculator, okay, like it said on the previous slide, no calculator. The only thing you are given is this periodic table and nothing else. It gives you the atomic number, it gives you the atomic symbol, okay, and it doesn't give you the name like it says over here. It will not tell you what element is what. You're supposed to know that, so you should recognize that that's iron, for example, and SN is tin, and W is tungsten, and just be very, very familiar with this, where the metals are, where the non-metals are, where the halogens, the alkali metals, right? We got our noble gases and so forth. And we're gonna look at this a little closer. So let's just start talking about what is chemistry. And chemistry is the study of matter. And so over here I have solid, liquid, and gas phases for you just to get you opening up here. Matter comes in all 3 phases, right? But also matter is made up of atoms that then build up and become molecules. Those molecules can be compounds and so forth, all right? So chemistry also deals with whether or not it's quantitative or qualitative, okay? So they're both in chemistry. What this basically means is that measurements are done, okay? So in other words, that you can measure it with a quantity, some sort of number, okay? So that's what quantitative means. Qualitative means that we have to describe what exactly we're seeing. So let's say the solution is 500 milliliters, okay? And then we have it here in this round bottom flask, not used for measurement. We have to describe what this is, okay? So the solution is pink. The solution is clear, okay? Originally, maybe it's clear to see through, right? It's transparent, I should say. And other things that you might have noticed. You can note that well, I want to say the temperature, but then that would be quantitative. Okay, but we're looking at two types of data here, and so the reagent was brown, and so forth. Okay, let's take a look at how substances are broken down. And so very nicely put here, we have matter, okay? And matter could either be a pure substance, or it could be a mixture. All right, now breaking them down even further, the pure substance was the periodic table of elements that we saw before. All right, and then sometimes those elements form compounds or molecules together. All right, and then we have the two types of mixtures. We have the homogeneous mixture and the heterogeneous mixture. The big difference between the two of these is that the homogeneous mixture is the same throughout. Okay, so this is the same. Okay, we're going to say throughout. While the heterogeneous mixture is different throughout. A great example of a homogeneous mixture would be milk. Milk is homogenized, right? It says the word homogeneous on it. It's a mixture of many things. It has fat, it has water in it, it has proteins in it, okay? And so that has been blended in a certain way where it's the same throughout. It is homogenized. On the other hand, sometimes things are different throughout, like a salad, right? Or maybe some, if let's say I put sand and water in the same glass, right? That's a heterogeneous mixture because the sand will go to the bottom and the water will be at the top. You can see right away that it's not the same throughout, no matter how much you shake it. Okay, eventually it'll settle down. Okay, milk doesn't settle down like that. Another example of a heterogeneous mixture would be ocean water or even the air we breathe. It changes all the time. It's different throughout. Okay, so keep that in mind. And this is nice. Compounds, for example, would be has to be two or more elements. So sodium chloride would be a great example, right? Or that's table salt. Okay, another example of a compound would be something you remember from biology. Glucose. Okay, great example of an element would be oxygen. Right? If it has the two there, it's still fluorine or hydrogen gas. These are all great elements, or even gold. That's the pure element, the pure substance. Okay, this is great. And so let's take a look at the next.